All right, friends. So we are finally in Unit 2, Humans versus Bacteria, still in our antibiotic resistance 5E, but we are on our elaborate, which means that there is a performance task coming up as it pertains to this 5E sequence. And guess what? This is officially the halfway point of Unit 2, believe it or not. So Unit 2 is kind of moving, right? It's been a lot of information, but very interesting, at least to me, hopefully to you as well. So our aim says, what are some ways we can kill harmful bacteria? Okay, so if we look at our do now, this is a graph in do now. It says plot the data from the table, surround each point with a small circle and connect the points. And then two says mark an appropriate scale without any breaks on each axis. And just wanted to throw in a do now that really emphasizes graphing, right? We want to make sure that we're practicing those skills. There's been a lot of like do nows that involve writing, but we want to make sure that we're practicing these skills of graphing as well. So let's just move our little, color wheel out of the way. And let's take a look at our data. All right, so on our x-axis, we have days, and on our y-axis, we have number of microorganisms in sample, okay? Now for me, I don't know about you, but I always start at 0, 0, the point of origin. As many times as I can, I always start there because then it gives me a better way of trying to find my interval. And on my x-axis, I have how many days? I have days 0, 1, 2, 3, 4, 5, 6, 7 days. But I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 hash marks that do not involve 0. So not including the 0 hash mark on the x-axis, I have 15 hash marks going across. But I already have 0 marked. So usually how I find my scale is that I take the number of hash marks that I have and divide it by my highest number on that axis. Now I know that 7 and 15 are not best friends, but they're friends. I know that 7 and 14, however, are best friends. And I know that 7 goes into 15 twice. So for every 2 hash marks, I'm going to mark off as 1. So this is effectively going up by 0.5, right? So we have 0, 0 0.5, 1, 1.5, 2, 2.5, 3, 3.5, 4, 4.5, 5, 5.5, 6, 6.5, 7. So no matter what, I know that all of my data on my x-axis for my days are going to fit on the x-axis. And I want to do the same thing for the y-axis, but we're not working with such nicer numbers. So not including 0, I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 hash marks going up. And then I have the number of microorganisms in a sample being the highest is 1,000, both at 0 and at 7, right? But I have 11 going up. So I can literally just go by 100s, right? If I go by 100s, then I know that I'll make it up to at least 1,000, with 1,100 being at the top, all right? But because these numbers are so big, I like to just count the number of hash marks and only mark off like some of them. So I'm only going to mark off where I see 500 and 1,000. The first one is 100, 200, 300, 400. I'm just going to mark off 500, 600, 700, 800, 900, and mark off 1,000, right? With 1 to spare. So now I know all of my data is going to comfortably fit here, where I'm going to be able to analyze and see trends and all of those type of things. And the reason why using an important scale is important, or is going to be of a benefit to you, is so that way all of your data is not just like marginalized or squished in like one area where you can't see trends or you can't see relationships or you can't make inferences. We want to be able to see the graph spread out so that way we can actually analyze it. And it's the same thing that scientists actually do. All right. So at day 0, the number of microorganisms in my sample was at 1,000. So if I just go over on my x-axis at 0 and then go up to 1,000, I'm going to mark a dot there. And again, this is going to be a line graph. I'm going to plot and coordinate my points. Now, my next one on day 1 is that it's at 500. I'm going to go over to 1 and then go up to 500. Then I'm going to go over to 2 and mark 100. And it makes it so much easier to graph when you actually have workable numbers instead of very awkward scales or awkward numbers with decimals and stuff, which sometimes is unavoidable, but in this case, it's working out in our favor. Now, 3, we have it going up by 100, but I know that the halfway point between 0 and 100 is 50, so I'm just going to mark it at the halfway point that's there. For 4, it's 40, which is going to be slightly lower than 50, obviously, but definitely above 0. And then at 5, we now see that the number of microorganisms in the sample has now risen on day 5 to 200. So it took a harsh dip and dive, and now we see that the population size is starting to increase over time as well. At 6, it goes up to 500. And at 7, it goes back up to 1,000. So in 7 days, we saw the population decrease from 1,000 at its highest peak down to 40 at its lowest peak on day 4, and then back up to 1,000 within 3 days. All right. Now I just need to connect the points using a straight edge. I'm making sure that these lines are straight. You're going to do the same. And again, this is going to be done in pencil. We never do graphs in pen, always in pencil. Okay. Just going to connect each point. Okay. And I always make it a habit to follow the example and circle the points that I've plotted. And voila. Hopefully you guys got something that was very similar as well. All right. So part A, antibiotics in action. Have you ever wondered why bacteria can become resistant to antibiotics? Well, bacteria go through the process of natural selection quicker than other species because of their fast reproduction cycle. This means they also adapt faster to different environments. In each population, some bacteria carry genes coding for antibiotic-resistant traits. This is just an overview, which we've already spoken about. Some in a population will naturally be antibiotic-resistant. Some bacteria produce their own antibiotics naturally in an attempt to kill other bacteria, especially when there is a lot of competition for resources in a certain area. So if you remember from the caves example, those cave-dwelling bacteria, they were producing antibiotics to fight off other bacteria in that given area, right? Because they wanted to outcompete them for resources. Other organisms, such as fungi, also produce antibiotics in order to fight off infections. So you guys probably don't know this, but the majority of our antibiotics that we take or that are prescribed by the doctor are primarily produced by fungi, mushrooms, mold, those type of things. When a bacteria population is exposed to antibiotics, the one with the advantageous trait will survive and reproduce, while the sensitive, those missing the trait, will die. That's natural selection. But what are these traits that bacteria have? Before we need to understand how bacteria or how antibiotics work, rather. So, antibiotics in action. Some antibiotics, for example, B-lactams, most known as penicillin. I bet you guys have never heard of penicillin before, or maybe you have. You're probably taking it before. Interfere with cell wall formation. So when the bacteria tries to replicate, the cell wall is destroyed and the bacterium dies. I bet you didn't know that some bacteria also had cell wall just like plants do. Other antibiotics, for example, quinolones, interfere with DNA and or protein synthesis in the bacteria, causing cell death. So some antibiotics literally interfere with the cell wall formation, right? Kind of punches holes in the cell wall physically. And then some stop DNA and protein synthesis from occurring in the bacteria, causing them to die. Antibiotics are a great strategy since they only target bacteria and not human cells. So that's a good thing, right? They only target. Yay. For this reason, antibiotics have been used for almost a century to treat bacterial infections, right? We calculated that like 80 plus years they've been used at least on farms, and for almost a century, they've been used to treat bacterial infections, right? Which means that they are effective, right? We still use them to this day. Now, unfortunately, some bacteria possess specific traits that allow them to survive the antibiotics. For example, some bacteria can destroy the antibiotics once they enter the cells using specific enzymes that inactivate the antibiotics. So the antibiotic goes literally inside of the bacteria, and the bacteria has a mechanism that has specific enzymes that literally inactivate that antibiotic and prevents it from killing the bacteria. Other bacteria have a different trait and are able to survive it by spitting out the antibiotics using special pumps on their membranes called efflux pumps, right? So as soon as it goes in to the membrane, it gets spit right back out, right? Almost like if you vomited bad food. It is important to remember that in each bacteria population, there are few bacteria that possess these traits and so can survive in the presence of antibiotics. When a patient is sick and treated with antibiotics, the bacteria possessing the resistant trait will survive and take over, creating a population of antibiotic resistant bacteria, right? So even though antibiotics have a lot of pros, they have different ways of killing the bacteria. Some bacteria have different mechanisms of making sure that they actually are able to resist and survive and reproduce. Okay. Now, how antibiotics work. Gee, I wonder who drew this beautiful illustration. Let's go over how do antibiotics work, right? So if we look at the legend, we see here that those little dots up here represent the antibiotic. Okay, now if we look at number one, we say that the sick person takes an antibiotic. I don't know why I'm obsessed with making the person's face look evil, but it's kind of funny, kind of camp, right? So the sick person takes the antibiotic, and then that leads to number two, right? Which, if we zoom in, the antibiotic that was swallowed goes towards the bacteria, and the bacteria has its cell membrane, it has a cell wall. It looks a lot like a plant cell, if you will. But this one is non-resistant, right? And if it's non-resistant, that means it's on its way to dying as a result of this antibiotic, right? If we zoom in on a portion of what's actually going on, right? We have the cell wall. Oops, let me use a different color. We have the cell wall, which is here. We have the cell membrane, which is here, right? That antibiotic is essentially going to kind of punch a hole through the cell wall, right? And as the antibiotic approaches the harmful bacterial cell, it destroys the cell wall and the cell membrane upon entry. And if we zoom back out, what that leads to is due to the destruction of the cell wall, cell membrane, and other organelles, the harmful bacterial cell can no longer reproduce. As a result, it cannot pass on its genes and it dies, right? So things are going to just start to maybe kind of leak out, right? That's not great for the bacteria. Leading to 5, which is I feel so much better. This person feels so much better. Why? Because the antibiotics are working. It's killing these bacteria, right? So that's great. Now, if we look at number 1, it says what organelles and bacteria are primarily affected as a result of antibiotic treatments. So primarily, first and foremost, is the cell wall. What's also affected is possibly the cell membrane. And if we remember from the reading, it says that some bacteria, or sorry, if we remember from the reading, it says that, see this, that these bacteria, like the way that antibiotics work is that they can destroy the cell wall or they can prevent or interfere with DNA and or protein synthesis. And if I'm talking about protein synthesis, then I definitely know that the organelle that's responsible for that are the ribosomes. So they can interfere with ribosomal function, which is not great. Any organism on Earth that has ribosomes that don't function properly will die. From bacteria to humans, that's always going to be true. We need our ribosomes because we require our proteins. They do so much in the body. All right. Now, that's how antibiotics work. Now, if we look at the second depiction, this is antibiotic resistance. So this is when it's not working. So as usual, the sick person takes an antibiotic, apparently an evil person, right? Which leads to number 2, the antibiotic approaches the harmful bacterium. And if we zoom into but this bacteria, I forgot to mention, this bacteria is resistant, unlike the one here, which was non-resistant. This one is resistant, okay? And just showing one way, right? The antibiotic was able to damage the cell wall, which is what we see here, and the cell membrane. However, once it entered, right, a receptor is going to take in that antibiotic and it's going to bind with it, leading to the efflux pump that they spoke about, spitting the antibiotic back out of the cell. And simultaneously, while it's spitting it back out of the cell wall that was damaged is able to start repairing itself along with the cell membrane, right? So when it's spitting it back out, it's going to be able to repair itself so that way its don't leak out like here or where it was permanently broken, it's going to start to repair while spitting it out, right? So if we read the caption for 4, it says the efflux pump embedded in the membrane quickly retrieves the antibiotic via a receptor molecule. This causes the pump to open up and eject or spit out the antibiotic before it can cause more damage. The bacterium is able to repair its membrane and cell wall. And as a result, since it didn't die, what is it going to be able to do? The harmful bacterium is able to survive and reproduce, passing on the gene for resistance to future generations. The sick person becomes more sick, right? So we see a whole bunch of resistant bacteria that are nice and evil, and the person is saying, I feel so much worse. Yeah, you bet you do, because we're overusing these antibiotics according to science. Now, number 2, what is the function of the efflux pump? Well, it says to eject, right? Which means spit out an antibiotic that has entered the bacterium, right? So the efflux pump is really important to a lot of these bacteria because this is their primary way of resisting the antibiotic. All right. So the efflux pump is going to be really important. It's going to come up again, I promise. Now, number 3 says, why does the sick person become sicker when bacteria build up resistance to antibiotics? Well, that's because they are able to, you guessed it, survive and reproduce, thereby passing on favorable traits of resistance to future offspring. This will create a population of antibiotic resistant bacteria. That's why the person got more sick, because now those bacteria are not being killed. They're resistant to the antibiotic and their medicine is not working. So the bacteria are really disrupting homeostasis. Bacteriophages. I'm going to say that this is going to be really important. Trust me, this will come up literally very soon. Let's read about bacteriophages. Sounds interesting enough. Have you ever caught a cold? If your answer is yes, it means a virus infected your cells in your throat and made you sick. So we're not even talking about bacteria anymore. We're talking about a virus. Did you know that there are some viruses that can only infect bacteria? Oh my gosh. Wait, so you mean to tell me that viruses also infect bacteria, not just humans, not just animals, not just plants, not just fungi, but literally these little microscopic bacterial invaders? Wow, let's deep dive into viruses. This seems like it's going to be cool. And that there's some that specifically only infect bacteria. That means I'm in the clear. Wow. They are called bacteriophages, or phages in short. Yes, Even bacteria can catch a cold. Phage comes from the Greek meaning to eat, and these viruses eat bacteria and kill them. Fortunately, they cannot infect human cells at all. This is a positive. I love this. They can't infect human cells at all? Great. Okay, let's read on. Billions of these phages are present all around us and protect us from bad bacteria. Hmm, seems like a smiley face to me. Let's continue. Scientists have used them even before antibiotics have been invented to treat bacterial infections. That's a net positive. So now we're using something that's like a virus, not like necessarily a medicine, but something that naturally occurs and we don't have to manipulate it. And we used it even before antibiotics. That's pretty incredible. Indeed, even today, you can buy them in a pharmacy in Russia. But how do these phages work? As you can see in the picture below, we don't have a picture below, but we'll draw one together, or I'll find a picture for you. Don't worry about it. They can attach to a bacterium and send their DNA inside. Once inside, the DNA will use the bacteria to make more copies of the phages, and eventually the bacteria will explode and release all the phages. Oh my gosh. So here's where we are, guys. Humans are smart. Bacteria are smarter. Viruses seem to be the smartest of all of them, right? Because they're literally outcompeting the smarter bacteria that are smarter than humans, and they leave us alone for the most part, right? They just want to destroy these bacteria. So they literally attach to the bacteria, they send whatever little DNA they have inside, they send it inside of the bacteria, and that DNA tricks the bacteria into making copies of these viruses. It hijacks the bacterial machinery to make copies of itself. And then once it's made as many copies as it can possibly make, it bursts from the bacteria, killing the bacteria. And then all of those other copies that are now replicated go and find more bacteria and do the same thing over and over and over again exponentially. That is fire, as the kids say. It's pretty fire. All right, the key, put that in air quotes, not air quotes, but quotation marks. The key these phages use to attach and enter the bacteria is the efflux pump on their cell wall. Oh my God. So the efflux pump that we were just talking about that spits out the antibiotic, that's the same place that these phages use to attach and enter the bacteria, right? So that's a trade-off for the bacteria to have an efflux pump. What's the benefit? It spits out the antibiotic. What's the cost? It makes them vulnerable to a viral infection and possibly being killed by these phages, these viruses. When phages infect a bacterial population, only those with a special trait will survive. In this case, bacteria with a non-functional efflux pump will survive since the phages will not be able to attach to it, while all the bacteria with a functional efflux pump will die, right? So the efflux pump is great. Why? Because again, it gets rid of those antibiotics that we are using to try and kill them. And it's saying, haha, you won't kill me now. But the phages are saying, haha, if you have an efflux pump, I can kill you, even though the antibiotics cannot, because I can attach to you, right? But if the bacteria doesn't have an efflux pump, then the viruses are like, oh man, I can't gain entry. I can't attach to it. I can't kill it. But what can kill it if they don't have an efflux pump to protect them is an antibiotic. Right? This means that if phages are used on a patient to get rid of a bacterial infection, only bacteria with non-functional efflux pumps will survive. This is, we're going to put a huge smiley face next to this because it seems as though these phages are our friends and we love this for them. Scientists are trying to use this cutting edge strategy to treat bacterial infections that are antibiotic resistant. What do you think are their strategies? How can scientists leverage phages to help treat antibiotic resistant infections? Right? The word leverage, the word leverage just basically means to use or it just basically means to maximize the advantage of something, if you will. Right? So how can scientists maximize their advantage of using phages to treat antibiotic resistant infections? Right? So this is important. We learn a lot. So part B, claim evidence and reasoning, our guiding prompt. How should we treat bacterial infections in the future? And this is actually going to be a homework assignment for you. We will not be doing this in class, right? So your question is, how should we treat bacterial infections in the future? Think cholera, right? Cholera seems to be antibiotic resistant. It keeps coming back again and again and again and again and again. And we don't really want that. We don't want them to keep coming back again and again and again and again and again. So as a result, we need to see what we can do to save these people from this uh, recurring or subsequent invasion of cholera. So our claim 1 says take antibiotics, and claim 2 says use bacteriophages. Using the information from this lesson, previous lessons, investigations in lab and or videos that we have watched, choose one claim above as the solution to answering the guiding prompt question. There is no right or wrong answer. However, you must use scientific reasoning and evidence to support the claim you chose. Remember, this is all about claim evidence reasoning. You're going to you take a claim, right? Whether you choose claim 1 or claim 2, you're not using both, just choose one. And using evidence from everything that we've gone over in the unit so far. Anything that you can remember from the videos, anything that you can remember from the labs that we've done, disease transmission, simulating a bacterial infection, any of the readings that we've had about resistance in caves and bacteria being able to use their own antibiotics and these bacteriophages that we just spoke about, and the efflux pumps, all of those things. You're going to use scientific reasoning and evidence to support the claim that you chose. Right? So if I choose claim 1, I might say we should take antibiotics to treat bacterial infections in the future, and here is why. Blah, 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 blah. Or if I choose claim 2, we should use bacteriophages to treat bacterial infections such as cholera in the future, and here's why. Blah, 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 blah. Okay? And again, this is homework. I don't need an intro, conclusion, none of that stuff. You get like one little page to write. I'm just looking for your claim evidence and reasoning. And with that, that wraps up antibiotic resistance. All right. So make sure that you do your homework, and you do have a performance task coming up. You guessed it, another claim evidence reasoning performance task. So study up. You guys are absolutely amazing, and I'll see you next time. Bye.